Hello, I'm Damla Senol Cali, and thank you so much for joining to my talk. I'm a sixth-year PhD candidate in the software research group at Carnegie Mellon University, and today I will be discussing some of the work I have been doing during my PhD on accelerating genome sequence analysis via efficient hardware algorithm co-design. So genome sequencing, which enables us to determine the DNA sequence of an organism, plays a pivotal role in areas such as personalized medicine, outbreak tracing like COVID-19, and understanding of evolution. However, there is no sequencing machine that takes the long DNA as an input and gives the complete sequence as an output. Instead, sequencing machines extract small randomized fragments of the original DNA sequence. So let's look at the process for like start to end for the genome sequencing. It starts with sample collection. It can be saliva, it can be blood from an organism, let's say human. And then after we collect this large DNA molecule, it goes through a wet lab preparation process where we divide this large DNA molecule into chopped DNA fragments. And these chopped DNA fragments are the inputs to the sequencing machines. And these sequencing machines generate the sequence reads, so the ACGT representation of these DNA fragments. And then we use these sequence reads in order to perform genome sequence analysis. So based on, we have multiple sequencing technologies, for example, Oxford Nanopore, PacBio, or Illumina, or some others as well. And based on the sequencing device, we obtain either short reads or long reads. So when I say short reads, I mean just a few hundred base pairs. And by base pairs means you can think them as like characters, ACGTs. But we are dealing with a highly accurate read. So the error rate is like around 0.1%. On the other hand, for long reads, we have thousands of millions of base pairs at each read. However, we are dealing with noisy reads, and the error rate is around 5 to 10%. And as the sequencing devices becomes cheaper and more accessible, the amount of genomics data that's being generated gets much and much higher. So, for example, this is an example from Oxford Nanopore. So when the devices get like larger but also available, we are seeing a huge increase in the amount of genomics data that we are producing. Or this is an example from Illumina, for example. So even in 2014, for each year, we were being able to generate more than 18,000. So it is even higher right now in 2021. However, the compute power doesn't match up with this trend. So modern sequencing machines generate read fragments at an exponentially higher than prior sequencing technologies, and with their growth far outpacing the growth in computational power in recent years. So computation is a bottleneck here in genome sequence analysis as well, because it cannot keep up with the pace of improvement in the sequencing part. And also what we should expect in near future. So we will see an introduction of very small, portable, and cheap devices that we can plug into our mobile phones, as you can see here on the right-hand side. And the left upper corner is the current state. So we have Minayan from Oxford Nanopore, for example. They are like USB-sized devices where you can plug into your laptop and do your sequencing and do the analysis on your laptop. So what we will be seeing in the near future is, so not laptops, but even doing this process using our mobile phones only. So which means that everyone can perform sequencing on their own genome, for example, by themselves using their mobile phones. However, as you can predict, the challenge is how to perform the required analysis on our phones, which have even more limited resources than our laptops. So rapid genome sequence analysis is currently bottlenecked by the computational power and also memory bandwidth limitations of existing systems, as many of the steps in genome sequence analysis must process a large amount of data. And as I showed, this data is getting higher and higher. So this is the problem. What we need, so our goal is to accelerate genome sequence analysis by efficient hardware algorithm co-design. And to achieve this goal, our approach is first analyzing the multiple steps and the associated tools in the genome sequence analysis pipeline and exposing the trade-offs between accuracy, performance, memory usage, and scalability, and then co-designing fast and efficient algorithms along with scalable and energy-efficient customized hardware accelerators for the key bottleneck steps of the pipeline. So to this end, I will cover 4 of my works today. And let's start with the first one, which is the bottleneck analysis of long-read assembly. And we published this paper 3 years ago in Briefings in Bioinformatics Journal. So when I say analysis, what do I mean? So what I mean is how we will analyze these reads generated by the sequencing machines. So they can be analyzed following 2 different approaches. First one is read mapping, and it is the process of aligning the generated reads against a reference genome to detect sequencing errors and also variations in the sequence genome. And the second approach is de novo assembly, which is the method of combining the reads to construct the original sequence when a reference genome does not exist, or when we want to improve an existing reference genome, or when we don't know what is the actual organism for the reads that we sequence. So if we don't know the organism, we cannot talk about a reference genome, for example. And this first work I am explaining right now is focusing on genome assembly pipeline using long reads. And why? Because with the emergence of long read sequencing technologies like Oxford Nanopore or PacBio, de novo assembly becomes a promising way of constructing the original genome, since a long read can span an entire repetitive sequence, which we have a lot in human genome, for example, and enable continuous and complete assembly. So this was a challenge when we have only short reads, but with the emergence of long read sequencing technologies, now it is promising. So how we start the genome assembly pipeline. So in Oxford Nanopore reads, what we have as an output from the sequencing device is raw signal data that represent changes in electric current when a DNA strand passes through a nanopore. Nanopore name comes from that. So the pipeline starts with this raw signal data, and the first step is called base calling. And in base calling, we translate this raw signal output of Nanopore sequencing devices into bases, ACGTs, to generate the actual DNA reads. So it is important to note that here is base calling is the only step unique for Nanopore sequencing. And the rest of the steps that I will cover are applicable for both of the long read sequencing technologies. By both, I mean Oxford Nanopore and PacBio. So in the second step, we compute all pairwise read alignments between each pair of reads, and this step is called read-to-read overlaps because of that. And in the third pipeline step, which is called genome assembly, which is the actual step, we generate an overlap graph using the overlap information from the second step, where each node denotes a read, and each edge represents the overlaps between the corresponding 2 nodes. So if 2 nodes, meaning 2 reads, have an overlap, then we have an edge between those 2 nodes. So we then traverse this overlap graph, produce a layout of the reads, and then constructs a draft assembly. So this draft assembly might have a lower accuracy than expected. So to increase the accuracy of the assembly, further polishing, or it's also called post-assembly error correction, may be required, but it's optional. So in the fourth step, which is the first optional step actually, we are mapping the original base called reads from the outputs of the first step to the generated draft assembly from third step. And we perform read mapping here. And in the fifth and final step of the pipeline, we polish the assembly with the help of mappings from the previous step and obtain the improved assembly. But as I said, those last 2 steps are optional if you want to increase the accuracy of the assembly. So in this work, what we did is we analyzed the tools associated with the multiple steps in genome assembly pipeline in terms of accuracy, speed, memory efficiency, and also scalability. And we revealed bottlenecks and trade-offs that different combinations of tools and different underlying systems lead to. And this is the first study on bottleneck analysis of Nanopore sequence analysis pipeline on real machines. And we first provide guidelines for practitioners such that they can determine the appropriate tools and tool combinations that can satisfy their goals. And also we provide guidelines for tool developers such that they can make design choices to improve current tools or future tools. So in this work, for accuracy and performance evaluations of different tools, we use 3 separate systems with different specifications. So we use the first system for obtaining the outputs for each data and for performing the accuracy analysis. And we choose the second system, which we call desktop, as it represents a commonly used desktop server. And the third system, which we call as Big Mem because of its large memory capacity and high number of cores, so it represents the opposite of a desktop server. So system 2 and system 3 are representing 2 like opposite configurations. And we use those 2 systems for our like scalability or memory or performance, like speed up kind of metrics. So when we look at our metrics, in terms of accuracy metrics, what we use is average identity, coverage, number of mismatches, and number of indels. So what we did is we compare each draft assembly generated after the assembly step and the improved assembly generated after the polishing step with the reference genome, and find out these accuracy metrics. And for performance metrics, we analyze the performance of each tool by running the associated command line of each tool with the user bin time command and also with perf. So we have 21 observations in our paper, but I will only highlight 3 key findings we made. So first, as I introduced, laptops are becoming a popular platform for running genome assembly tools, as the portability of a laptop makes it a good fit for an in-field analysis, because as I showed, now we have devices which we can plug into our laptops and perform sequencing. However, we are dealing with greater memory constraints, lower computational power, and also limited battery life. So the second finding is memory usage, of course, is an important factor that greatly affects the performance and also the usability of the tool. So data structure choices that increase the memory requirements, or algorithms that are not cache efficient at all, or the algorithms that don't keep memory usage in check with the number of threads, were suffering because of bad memory usage or inefficiency in terms of that. And third, as expected, again, scalability of the tool with the number of cores or threads is a very important requirement, because we are dealing with very time-critical analysis here. However, we also observe that parallelizing the tool can increase the memory usage. So when we don't divide input data into batches, or when we don't limit the memory usage of each thread, or when we divide the data set instead of the computation between simultaneous threads, then we are affecting the scalability of the tool, also ending up with an increase in the memory usage. So our goal should be first, obtaining high performance while maintaining low power. Second, memory efficiency. And third, scalability or high parallelism. So to meet these goals that we just discussed, we need to co-design hardware and software. And in our second work, GenASM, we are proposing a hardware-software co-design for genome sequence analysis. As I mentioned, read mapping is one of the steps of the assembly pipeline, but also, if you remember, it's also another main approach to perform genome sequence analysis. And it's a very commonly used pipeline. And one of the very common downstream analysis after read mapping is variant calling, for example. So it's a very crucial step. And let's look at the read mapping pipeline in detail. So we can see it as a 4-step process, and it's also sometimes called seed and extend process. So first, read mapping starts with indexing the reference genome, and this index contains all possible fixed length substrings, or we call them sometimes k-mers, as the keys, and the exact match locations of these substrings in the reference genome as values. So this is a pre-processing step only done only once for each reference genome. So we obtain a hash table-based index, which we will use for the next steps. So in the second step, seeding. So in this step, we query the index structure for determining potential mapping locations of each read in the reference genome using the substrings, or we call them seeds, so that's where the name of the step is coming from each read. And in the so we end up finding potential mapping locations for each read. In the third step, pre-alignment filtering uses filtering heuristics to examine the similarity between every read and its potential matching segment on the reference genome that are identified in the second step. And goal here is to eliminate most of the dissimilar sequences to decrease the number of required alignments. And in the fourth step, which is the most expensive step of the pipeline, for all of the non-filtered candidate mapping locations, read alignment, or it's also called seed extension, performs a dynamic programming-based alignment to determine the best location of the query read. And also as part of the step, traceback operation between the reference segment and the query read is performed in order to find the optimal alignment. So in read mapping, we take each read, align it to one or more possible locations within the reference genome, and find the matches and differences between the read and the reference genome segment at that location. And as I showed, multiple steps of read mapping must account for the sequencing errors, as I explained at the very beginning. So in short reads, we have less percentage for these sequencing errors, but when we move to long reads, we are dealing with 5 to 10% of sequencing errors. And also we have differences caused by genetic variations. So as you can imagine, all of us have different genomes. Yes, they share a large portion of common sequences, but we all have genetic variations which cause differences in our sequence genomes. So as a result, read mapping must perform approximate string matching, or ASM, in order to account for these differences and sequencing errors. However, it is one of the main contributors of the bottleneck that we identified, which is computational power and memory bandwidth limitations of existing systems. So our goal in this work is to design a fast and flexible framework for both short and long reads, which can accelerate multiple steps of genome sequence analysis. So to this end, we propose GenASM, and we base GenASM on BiTAP, which is an ASM algorithm that uses only fast and simple bitwise operations, making it amenable to efficient hardware acceleration. And to our knowledge, GenASM is the first work that enhances and accelerates BiTAP. So we modified BiTAP to support long reads and also to enable parallelization. We also developed a novel BiTAP-compatible algorithm for traceback, and we co-designed specialized hardware for both algorithms. Let's look at approximate string matching and the BiTAP algorithm in detail. As I introduced, the reads may not exactly map to the reference genome due to genetic variations and sequencing errors. And as we can see from this example, these errors and differences, which are also known as edits, can take the form of deletions, substitutions, or insertions. So we need approximate string matching, and the goal of approximate string matching is to detect these differences and also similarities between two sequences. And in genomics, ASM is required to determine the minimum number of edits between two genomic sequences, and this is called edit distance, and also to find the optimal alignment with a traceback step, where we find the sequence of matches, substitutions, insertions, and deletions along with their positions. So for example, in this example, the optimal alignment would say, okay, for this reference segment and this read, we have 3 matches followed by a deletion. We have 6 matches followed by a substitution, and for the substitution, G is substituted with a C, and 6 more substitution, and we observe an insertion, and the inserted character is T, and we observe 2 more matches. So this is the optimal alignment information that we have to obtain after a traceback step. So approximate string matching is usually implemented as a dynamic programming-based algorithm. However, it has quadratic time and space complexity, so it is desirable to find lower complexity algorithms for ASM. And one candidate to replace DP-based algorithms for ASM is the BiDAP algorithm. And BiDAP, as I said, uses only fast and simple bitwise operations to perform ASM, so it is amenable to efficient hardware acceleration. And BiTAP tackles the problem of computing the minimum edit distance between a reference text, such as a reference genome, for example, and a query pattern, such as a query read, with a maximum of k many errors. So the algorithm has two steps. In the pre-processing step, we generate one pattern bitmask for each character in the alphabet. Here, the alphabet is ACGT, so only 4 characters. And these pattern bitmasks help us to represent the query pattern in a binary format. And in the second step, which is called searching or edit distance calculation, this is the main step of the algorithm. Here, we examine each text character one by one by using the pre-processed pattern bitmasks, status bit vectors, which are updated at each text iteration and hold the partial match information for the text characters examined so far, and also simple bitwise operations. So we find that BiTAP has 5 limitations that hinder its applicability and efficient hardware acceleration for genome sequence analysis. Let's look at the first one, which is data dependency between large number of iterations. So first, BiDAP iterates over each text character as the outer loop and iterates over each possible edit distance 1 to k, because as I said, k is the maximum allowed number of errors. So the inner loop iterates from 1 to k as the inner loop. So when we have long error-prone reads, both of these loops need to iterate over very large numbers. And on top of this, there's 2-level data dependency in BiDAP. So there's an outer loop data dependency when computing the deletion, substitution, and match bit vectors, as they require old R. And these old R bit vectors coming from previous text iteration. And also, there's an inner loop data dependency when computing the insertion bit vector, as it requires RD-1, and this R is coming from the previous inner loop iteration. So let's look at the second limitation of BiTAP, which is no support for traceback. So BiTAP generates these 4 intermediate bit vectors, deletion, substitution, insertion, and match, but they are not stored or processed with a traceback step to find the optimal alignment in the baseline algorithm. And also, on top of these 2 algorithmic limitations, we find one more, which is no support for long reads. So in BiTAP, the query length, so the read length is limited by the word size of the machine running the algorithm, because each bit vector, it can be either pattern bitmask or the regular status bit vectors, has a length equal to the length of the pattern, and we perform the bitwise operations on these bit vectors. So the algorithm does not support long reads whose lengths are on the order of thousands to millions of base pairs. And these are the algorithmic limitations of the BiDAP algorithm. Even after we solve these algorithmic limitations, we find that we cannot extract significant performance benefits with algorithmic enhancements alone. For example, while BiTAP iterates over each character of the input text sequentially, we can enable text-level parallelism to improve its performance. However, the achievable level of parallelism is limited by the number of compute units exist like in an existing system. And so we would expect that a GPU, which has thousands of compute units, can overcome the limited compute parallelism issues that CPUs experience. However, we find that a GPU implementation of the BiDAP algorithm suffers from the limited amount of memory bandwidth available for each GPU thread. And these cover the hardware-related limitations. And in order to overcome these limitations and design an efficient accelerator, we find that we need to both modify and extend the BiTAP algorithm, and on top of that, develop specialized hardware that can exploit the new opportunities that our algorithmic modifications provide. So to this end, we propose GenASM, an ASM acceleration framework for genome sequence analysis. And to our knowledge, GenASM is the first ASM acceleration framework for genome sequence analysis, and it is also first that enhances and accelerates BiTAP. And we make significant modifications to BiTAP in order to overcome those 5 limitations I just explained. First, we modify the BiTAP algorithm by extending it to support long reads and by eliminating loop carry data dependencies to enable parallelization. Second, we develop a novel BiTAP-compatible algorithm for traceback, which uses information collected during ASM about the different types of errors to identify the optimal alignment of reads. So these are the algorithmic contributions of GenASM. And third, we co-design specialized low-power and area-efficient hardware for both algorithms, and this covers the hardware-level contributions of GenASM. So let's look at the hardware design of GenASM. So our co-design hardware consists of 2 components. First, GenASM DC, which provides hardware support to efficiently execute our GenASM DC algorithm, which is the modified BiTAP algorithm that performs distance calculation. And second, GenASM TB, which provides hardware support to efficiently execute our new traceback algorithm to find the optimal alignment. GenASM also has 2 types of SRAM buffers, DC SRAM and TB SRAMs. So let's look at the high-level overview of GenASM. So GenASM execution starts when the host CPU issues a task to GenASM with the reference and the query sequences locations. Then GenASM DC reads the corresponding reference text region and the query pattern from the memory, and then writes these to its dedicated SRAM, which is DC SRAM. And in order to decrease the memory footprint of the algorithm, GenASM follows a divide and conquer approach, where we divide the text and pattern into overlapping windows and perform the traceback computation for each window sequentially. And after all of the windows' partial traceback outputs are generated, we merge them to find the complete traceback output. Because of that, as the next step, GenASM DC divides the reference text and the query pattern into multiple overlapping windows. And for each subtext and subpattern, GenASM DC searches for the subpattern within the subtext and generates the bit vectors. And GenASM DC writes the generated bit vectors to TB SRAMs. And once GenASM DC completes its search for the current window, GenASM TB starts reading the stored bit vectors from TB SRAMs and generates the window's traceback output. Once GenASM TB generates this output, then GenASM DC computes the next window and repeats from steps 3 to 7 until all windows are completed. So our specialized compute units and on-chip SRAMs helps us to first, match the rate of computation with memory capacity and bandwidth. Second, achieve high performance and power efficiency. And third, scale linearly in performance with the number of parallel compute units that we add to the system. Let's look at GenASM DC's hardware design. So we implement GenASM DC as a linear cyclic systolic array-based accelerator using small and very simple logic components. And this design helps us to maximize parallelism and minimize memory bandwidth and footprints. So processing core is the basic compute component, which computes the intermediate bit vectors, deletion, substitution, insertion, and match. And when we add the flip-flop based storage logic around the processing core, we define a processing element, or PE. And multiple PEs are concatenated to define a processing block. We also have DC SRAM, which stores the reference text and the pattern bit masks for the query read. And we also have TB SRAMs, which store the intermediate bit vectors generated by each PE of GenASM DC for later use by GenASM TB. And we implement GenASM TB hardware using very simple logic, which first reads the bit vectors from one of the TB SRAMs using the computed address. Next, performs the required bitwise comparisons to find the traceback output for the current position, and then computes the next TB SRAM address to read the new set of bit vectors. So it goes back to step 1. After GenASM TB finds the complete traceback output, which is also called cigar string, it writes this output to main memory and completes its execution. So we demonstrate the efficiency and flexibility of the GenASM acceleration framework by describing 3 use cases of approximate string matching in genome sequence analysis. First, the last and most expensive step of read mapping, which is read alignment step for both short and long read mapping. Second, pre-alignment filtering for short reads. And also third, edit distance calculation between any 2 arbitrary length sequences, and this is one of the fundamental operations in genomics that measures the similarity or distance between 2 sequences. And we believe that GenASM framework can be useful for many other use cases, and we discuss some of them briefly in our paper, including generic text search, for example. So let's look at our evaluation of the GenASM design and framework. So we will evaluate GenASM using a combination of synthesis of our DC-MTB accelerator data paths and detailed simulation-based performance modeling. And we evaluate a 16 gigabytes of HMC-like 3D stack memory architecture with 32 volts. So our accelerators can be ported anywhere in the system, but in order to achieve high parallelism and also low power consumption, we place both GenASM DC and TB in the logic layer of 3D stack memory. And as I will show next, our hardware is highly efficient in terms of both area and power, so they are within the budget limits of the logic layer. For the use cases that we evaluate, we compare GenASM with state-of-the-art software and also hardware baselines. So I won't go into details for the evaluation methodology of each case, but you can refer to our paper for more details. So let's look at our key results. We synthesize our DCM-TB accelerator data paths with a typical 28 nanometer process. And when both accelerators operate at 1 gigahertz, we find that for 32 GenASM accelerators, so one for each HMC volt, total area overhead is 10.69 millimeters square, and total power consumption is 3.23 watts. And we observe that both area and power consumption of GenASM at a single volt is around 1% of the area and power consumption of a single Xeon CPU core. So GenASM has low area and power overheads. Let's look at our results for the first use case, read alignment. So as software baselines, we compare GenASM with BWA-MEM, which is shown with red bars, and Minimap2, which is shown with purple bars. And both BWA-MEM and Minimap2 are commonly used and state-of-the-art read mappers. And we find that GenASM achieves 648 times speedup over 12-thread run of BWA-MEM and 116 times speedup over 12-thread run of Minimap2, while reducing power consumption by 34 times and 37 times. And compared to Gacktov-Darwin, which is a state-of-the-art long-read alignment accelerator, GenASM provides 3.9 times better throughput, 6.6 times better throughput per unit area, and 10.5 times better throughput per unit power. And for short reads, compared to the same software baselines, GenASM provides significant speed up and reduction in power consumption, which is similar to the trend we observed in long reads. And compared to Scylla-X of GenAX, which is a state-of-the-art short read alignment accelerator, GenASM provides 1.9 times better throughput while using 63% less logic area and 82% less logic power. Let's briefly look at our results for our second use case, which is pre-alignment filtering. Here is the takeaway. So compared to SUJI, which is a state-of-the-art FPGA-based filter, GenASM is more efficient in terms of both speed and power consumption, while significantly improving the accuracy of pre-alignment filtering. And when we look at our third use case, edit distance calculation, our key takeaways are, compared to software baseline, EDLib, GenASM provides 2 to 4 orders of magnitude speed up while reducing power consumption by 2 orders of magnitude. And compared to the hardware baseline ASAP, GenASM provides 9.3 to 400 times speed up while consuming 7 times less power. And before I summarize GenASM, here is a list of additional details that you can find in our paper. To summarize, in this work, we focus on approximate string matching because it is one of the main bottlenecks of genome sequence analysis. We propose GenASM, which is an approximate string matching acceleration framework to accelerate multiple steps of genome sequence analysis, and GenASM is the first work that enhances and accelerates BiTAP for ASM with genomic sequences. And in GenASM, we co-design our modified, scalable, and memory-efficient algorithms with low-power and area-efficient hardware accelerators. We evaluate 3 different use cases and show that GenASM is significantly more efficient for all these 3 use cases in terms of throughput and throughput per unit power than state-of-the-art software and hardware baselines. So next, I will talk about 2 ongoing works where we build upon GenASM. And the first one is BitMac, where we propose the FPGA-based near memory acceleration of bit vector-based sequence alignment, which is with GenASM actually. So we implement and map GenASM DC and TB to an FPGA with HBM2 and demonstrate end-to-end application accelerator. So key idea here is HBM2 offers high bandwidth and FPGA resources offer high parallelism, so we can instantiate multiple copies of GenASM accelerators and obtain like an efficient prototype actually. However, our key findings are, due to the high amount of data needs to be saved for the TB part, we are bottlenecked by the amount of on-chip memory we have. So because of that, we cannot saturate the high bandwidth that multiple HBM2 stacks that are on package provide, because we cannot instantiate multiple, like as many BitMac or GenASM accelerators as we want to saturate that high bandwidth because of the limited on-chip memory we have. So we need either an algorithm level modifications to decrease the amount of data that need to be written as part of the TB process, or new FPGA chips, which has a higher amount of on-chip memory capacity and also bandwidth. So in our second ongoing work, we explore how we can utilize this bandwidth by also including another step of read mapping pipeline to our design. But also here in this work, we explore a similar but different genomics problem, which is called sequence to graph mapping. As you recall, in read mapping, we start with a linear sequence of a single reference genome. However, this linear reference genome does not represent the genetic variations exist in a population and cause reference bias when performing read mapping with respect to it. So we have a lot of sequence genomes, but when we do like linear or traditional read mapping, we are limiting ourselves with a linear sequence. On the other hand, we have a lot of variation information which we can use, but without using them, we have reference bias. So the genomics community has recently started gaining interest in genome graphs, which include the reference genome together with genetic variations and provide a compact representation of the reference genome. And these genome graphs enables us to move away from aligning with single reference genome and toward using the sequence diversity. So for example, this is one reference genome, let's say we have. So we start with a single node. So when we have a second reference genome, and here, as you can see, for the 4th position, instead of T, in this one, we are observing a G. What we can do, but they also share 3 length substrings and 4-length substring at the beginning and end. So we can divide this single node to 2 nodes where they represent the shared strings, and we can have a path with a single node in between, which is for the first reference, and we can have an alternative path which has G in the middle as another node. So we can with this graph representation, we can include both of these references. Let's say we also have an additional T as in the same position. So we observe an insertion here. What we can do, we can have an additional T node in between so that this would be an alternative path from T to ACGT actually. So we would represent the insertion this way. And in the fourth reference, let's assume we don't have any character in that position, meaning that it's a deletion. So what we need to do is from ACG to ACGT, we should have a direct path to represent this reference. So we can, with this compact representation, we can have both, like all four of these reference genomes. So we don't have to stick with only one of it. So, since traditional read mapping causes reference bias, aligning sequences to graphs emerged as a newer field, and practical tools only start to emerge, where most of them are specialized for one purpose only. So, hardware acceleration of sequence to graph mapping is an important but unexplored research problem. So, in GenGraph, our goal is to design an accelerated framework for sequence to graph mapping, which provides high performance and high accuracy. Also, it's important to be comprehensive in terms of genome graph types, sequencing reads, and also applications. To this end, we propose BetaLine, which is the modified version of GenASM algorithms and also hardware accelerators in order to perform sequence to graph alignment. So original GenASM algorithms and accelerators are for sequence alignment. So here in GenGraph, we modify it and come up with BetaLine, which is for sequence to graph alignment. And we also propose the first minimizer-based seeding hardware, which we call MinSeed. So this is for the seeding step of the mapping pipeline. And this module, like this accelerator, can be used for both traditional linear read mapping and also graph mapping. So let's look at overview, quickly go over the overview of GenGraph. So we have, again, have a host CPU, which will provide the query reads to us, like stream them. But here in main memory, what we have is, we both have the genome-based representation of our reference genome and also the preprocessed index for this reference. So these all are preloaded to the main memory. So what we have as part of MinSeed is, like our MinSeed accelerator, we have the MinSeed accelerator, and also we have two types of SRAMs. So first, host CPU streams the query read, and this MinSeed accelerator finds the minimizers for this query read and writes them to MinSRAM. So MinSRAM stores the minimizers for the current read. And then we access the main memory to obtain the locations of these minimizers using the preprocessed index. And then these locations are sent back to our accelerator to perform some simple filtering process. And then the non-filtered seeds are being written to seed SRAM. So seed SRAM has the selected minimizer locations, like the seed locations for the given query read. Then next, what we have is, for each of these seed SRAMs, like each of these seeds, we will send the surrounding subgraph that corresponds to the seed location to BetaLine. And in BetaLine, similar to GenASM DC, we have DC SRAM, which stores this subgraph that we will perform the alignment with, along with the query reads. And then we perform the modified DC operation, which I call as BetaLine DC. And then, again, the generated bit vectors are being written to TB SRAMs. And here, what is different is we also have hop queue registers in order to correspond to the the hops that we have as part of our graph. So we don't have just neighbor accesses when performing the alignment, but we can also get data from non-neighbor nodes as well because of the graph structure. So we, in order to take them into consideration when performing the computation, we also have hop-queue registers on top of the GenASM design here in GenGraph as part of the beta line design. And also similar to GenASM, we also have a beta line TB accelerator again, which performs the traceback operation. So we are still finishing the design and evaluation, but we hope to have more information to share in the upcoming months. So this covers the works that I want to present today. So to conclude, we observe that rapid genome sequence analysis is bottlenecked by the computational power and memory bandwidth limitations of existing systems, since we have to as part of many of the steps, genome sequence analysis must process a large amount of data. So we showed that genome sequence analysis can be accelerated by co-designing fast and efficient algorithms along with scalable and energy-efficient customized hardware accelerators for the key bottleneck steps of the pipeline. And to this end, in my first work, we aim to understand where the current tools and algorithms do not perform well in order to develop better tools and algorithms, and also to understand the limitations of existing hardware systems when running these tools and algorithms in order to design efficient customized accelerators. So for this work, our key takeaway is we need memory efficient, low power, and scalable designs for genomics in order to exploit the advantages that new sequencing technologies provide, such as near sequencing device analysis for real-time analysis, and portability to perform genome sequencing and genome sequence analysis anywhere in the world and even in remote places. And to this end, in my second work, we propose GenASM, which is an hardware algorithm co-design to accelerate one of the key bottlenecks of genome sequence analysis, which is approximate string matching. And in GenASM, we co-design our modified, scalable, and memory-efficient algorithms with low-power and area-efficient hardware accelerators. And we evaluate 3 different use cases and show that GenASM is significantly more efficient in terms of in terms of both throughput and throughput per unit power, and compared to the both state-of-the-art software and also hardware baselines. And in my third work, we propose an FPGA-like prototype for our GenASM accelerators, where we implement and map GenASM DC and TB to an FPGA with HBM2 and demonstrate end-to-end application acceleration. And we revealed that due to high amount of data needs to be saved for the traceback step, we are bottlenecked by the amount of on-chip memory we have, and we cannot saturate the high bandwidth that multiple HBM2 stacks that are on package provide. So in my fourth work, in order to exploit the high bandwidth that HBM2 provides, we propose the first minimizer-based seeding accelerator, which performs the memory access heavy step of read mapping, which is querying that hash table index. And in addition, instead of traditional linear read mapping, GenGraph focuses on graph mapping problem or sequence the graph mapping problem, which is a newer genomics problem, to get rid of the reference bias and provide a more comprehensive and accurate solution for one of the key genome sequence analysis steps, read mapping. And besides these works, I was also the co-author for multiple other works, which are from Software Research Group. Thank you so much for listening, and I look forward to providing more details regarding the ongoing projects we have in the upcoming months. Thank you.